Thank you all for attending the workshop today. So as Jonas said, we'd like to take the time before we get to the featured talks to go through all the innovations and new releases that PacBio has come out with since we've had a chance to update you at PAG 2024 last year. So first and foremost, I wanted to introduce the SPRQ Chemistry. So this is our first chemistry advancement on our Revio system, and there are three main features. The first is that we've decreased the DNA input requirement going into the annealing and binding process. So this will be great for smaller bodied organisms or organisms where it's hard to extract a lot of DNA. We've also increased the throughput by about 30%. So the average Revio cell yield is now 100 to 120 gigabases. And we've continued to innovate on the methylation side. So we improved the 5MC caller that's already on board, and we've also increased this to encompass 6MA to really enable our FiberSeq application. So digging into that a little bit more, we lowered the input amount, as I had previously stated, to 500 nanograms. This is a reduction from 2 micrograms previously. And so again, what this allows for is additional sample types and also not stressing you so much when it comes to acquiring enough DNA. I also want to stress that this is 500 nanograms going into annealing and binding. We can actually go into our library prep process with 50 nanograms. So if you do have a lot of small organisms that you're looking to sequence, but you can't even hit the 500 benchmark, you can now multiplex those together to hit 500 and then move forward with the sequencing process. And I don't want you to just have to take my word for it. So we partnered with Ellie and Luke Talon from University of Maryland. They were kind enough to provide us with samples that they had previously sequenced on the first iteration of the Revio chemistry. And we used a quarter of the DNA input and sequenced this on the new SPRQ chemistry. So you can see here that in all three of these organisms, there was a substantial increase in the amount of yield that was coming out on the back end. And if you take a look at the long-winged butterfly on the bottom there, you can see that this was actually utilizing our Kinnex RNA workflow. And so digging into that a little bit, if those of you that remember from last year's PAG, Kinnex is our RNA workflow, but it's also a way for us to concatenate molecules together and move into other amplicons such as 16S, et cetera. And previously, per Revio cell, we were able to get about 40 million full-length non-concatemer reads. If you utilize the SPRQ chemistry with our Kinnex kits, we can now build out enough synthetic molecules with the SPRQ chemistry to get 60 million reads per Revio cell. So this is a 50-fold increase in the number of those isoforms that you're able to see. And with the lower DNA input requirements, this also gives you additional experimentation flexibility because you can now run a Kinnex library across 4 Revio cells where previously you were only able to generate enough material to put that across 1 or 2. So that allows you more flexibility, barcoding at the cDNA level that you can then demultiplex later on. Just a reminder, our current recommendations are 10 million reads per tissue type as you're going to do an annotation. So on the V1 version of the chemistry, that allowed you to get roughly 4 samples per Revio cell, and with the SPRQ chemistry, we're increasing that recommendation up to 6. So again, what that looks like is that your species of interest and your different tissue types. Previously, you could multiplex 4 together, and now with the improvements, that allows you the flexibility to do 6. So whether that's 6 tissue types from the same organism, or if you're looking to do 3 from different organisms, now you can do 2 on the same cell. Also wanted to quickly call out the improvements that we've made on the methylation front. So as a reminder, as our polymerase is incorporating bases, there's going to be a lag in the kinetics as it's incorporating a specific base if that base is tagged with a motif. And so we improved the accuracy of our 5MC calling by about 10%, and we've also onboarded the 6MA caller to, again, really enable the FiberSeq chromatin assay that you see here on the slide. And these chemistries are now available. It's the same price per run as the additional chemistry, so if you're interested in bringing this into your lab, feel free to stop by the PacBio booth to learn more. Next, I want to spend a little bit of time walking you through the Vega systems and what the specs are on this benchtop sequencer. So this is really our solution to help decentralize our sequencing model and be able to bring our HiFi technology across the globe into smaller and medium-sized labs as well. So here's a peek of that system that you saw in the video that Jonas played. And if we get into the details a little bit, here's the benchtop footprint that you see the metrics for on the left. We also have put it into standard electrical as well as networking, so you no longer have to have any special plugs to bring this into the laboratory side, and we don't have a nitrogen requirement either. So it's just the Vega box and the standard plug that you need to bring this into your laboratories. Same technology that we utilize on our HiFi system, and then it has a very elegant design, and we've been able to maintain deep consensus methylation calling and direct cloud uploading that you're used to seeing on the Revio systems. And if we look at the specs in a little bit more detail, as a reminder, the Revio system can sequence up to 4 SMRT cells at one time. The Vega system does 1 SMRT cell at a time, and that 1 SMRT cell will give you about 50 to 60 gigabases per run. You have configurable movie times that you're able to select, so you can do a 12-hour or 24-hour movie, and we maintain a lot of the read length and accuracy that you're used to seeing from HiFi data. Here's a quick peek at the pricing. So list price for this system is $169K USD, and it equates to about $1,100 per sequencing run. And then we also have different programs available if you prefer to offload some of the CapEx into the consumable side as well. It's also extremely simple to load. We just have 2 consumables, one that is at room temperature, and then the sequencing plate that has the same storage conditions on the Revio side. These can be loaded within seconds, and then the run starts immediately as soon as that tray retracts. And again, I don't always want you to just have to take my word for everything. So we partnered with our friends at the CCGP out of UC Davis to secure some samples that were run through this biodiversity initiative that were sequenced on their Revio systems there. So you can see that the flannel bush, the rockfish, and our happy friend, the sea otter here, were all able to generate 64.9 gigabase yields on the Vega system. And then you can see the equivalent HiFi yields here on the slide. I did want to make a quick comment because it does seem suspicious that all 3 of these organisms got the exact same output. The reason for that is as we were miniaturizing the capabilities on the Vega system to actually do the analysis, currently that is the maximum yield that this system is able to output. So hopefully this is our way to really help standardize the output of these runs, but also this gives us a lever that we can pull in the future to continue to iterate as future chemistries come out on this system. We also went one step further with this experiment as well and went ahead and did the assemblies of these to compare to the Revio to prove that this is the same HiFi data you're used to seeing. And you can see here that in all 3 of these instances, the Vega data yielded relatively the same assembly metrics as the Revio data, and it was also able to phase into the 2 haplotypes that you see here. And if you were able to see Mike Schatz's talk yesterday, he has a great initiative called the Genomic Data Science Community Network, which is a really neat program where they're able to get students into sequencing soil metagenomic work. Mike previously had Revio data as well, and we were able to sequence one of these soil metagenomes on the Vega system for him prior to the PAG conference. And you can see here that on the metagenomic side, we also see very similar outputs to what the Revio system is capable of. And then finally, I wanted to touch on a few additional advancements that we made throughout the year as well. So first and foremost, in 2024, we launched the pan-DNA kits, which was really our way to simplify our nanobind extraction portfolio into a single kit. And not only did we turn 3 kits into 1, but we also onboarded additional sample types. So insect was the main one that we added into this suite, and we have now a lot of examples out there of utilizing the pan-DNA kit to successfully extract high molecular weight from insects. And as customers look to fill their Revio systems, we're also trying to make sure that we're advancing on the high throughput and automation front. So our nanobind CBB kits, as well as our high throughput 1 milliliter blood kit, We have automated through our PacBio compatible program with Thermo Fisher Scientific and Hamilton, and these protocols feed seamlessly into our nanobind extraction protocols. So again, if you're interested in bringing high throughput capabilities into your lab, feel free to stop by the booth and we're happy to talk about that in more detail. Jonas alluded to this previously, but we also announced today on our PacBio official blog that we're making improvements to our ultra-low input workflow. So this is very exciting for us because previously we weren't able to port this over to our Revio systems, but with the improvements we're making to this kit, it'll be available on Vega, Revio, and our Sequel IIe systems. We've also simplified the amplification, so you no longer have to do two separate amplifications. We've converted this into a single step. And we've reduced the input amount from 5 nanogram down to 1 nanogram. And then finally, we've increased the recommended genome size that this kit can support up to 3 gigabases. And Scott's going to talk about this a bit as well. So with the help of the protocol coming out of Megan's lab at UC Davis, he's also able to utilize a lot of these updates that we've made to the ultra-low workflow into doing HiFi technology on the PacBio systems. And finally, I wanted to call out quickly a lot of the bioinformatic analysis tools that we all have available through our suite in Bioconda and GitHub now. So we're adding new tools all the time, and these pages are curated very well and kept up to date. So if you're interested in checking out the tools, we have an expanded fleet that you see here in the structural variation space. And with that, I would like to hand it back over to Jonas. So thank you for your attention, and please enjoy the speakers that we have lined up for you. Thank you.